Now that we've finished our discussion of mitosis and then cancer, which is, of course, what happens when mitosis goes incorrectly and the cell loses control of that type of division, as you know from our last lectures, we want to move on to meiosis. And so meiosis has a very particular, specific purpose in biology. Meiosis only occurs to form the gametes, the eggs or the sperm. And so, of course, it would make sense that meiosis would have to cut the genetic material in half. We realize from our previous discussions that humans, like most other animals, are diploid. And that means we have two copies of each of our chromosomes. And so then, logically, the gametes, the eggs or the sperm, would have to be haploid, have one copy of each chromosome, and they would come together to make a diploid organism in the next generation. And so meiosis is, again, the specialized form of division that occurs only in the gonads to produce the gametes, the eggs and the sperm. It reduces the chromosomes in half in each cell. So chromosomes come in these things called homologous pairs that we're familiar with from previous discussions, but we realize that homologous pairs contain alleles. For instance, briefly, if we look down at the bottom, as I'm pointing to, of our screen, the bottom right-hand corner, the little green dot there would represent, these are two chromosomes, one from the father and one from the mother, in an adult individual. And the green dots would represent genes. For instance, the green dot could represent a gene that contributes to hair color. And this gene is possessed by both, it's on both the chromosome that came from the father and the chromosome that came from the mother. But those genes aren't necessarily the same. If there's a gene for red hair on the father's chromosome, the only thing we know for sure is that on the mother's chromosome there's also a gene that codes for hair color. It could be any color of hair. And so this is what we refer to as alleles, A-L-L-E-L-E-S, alleles, alternate forms of a gene. So in the case of humans, somatic cells, which mean the non-reproductive cells, have 23 pairs of chromosomes total. There are 22 pairs of autosomes, meaning 22 pairs of chromosomes that have nothing to do with sex determination, and one pair of sex chromosomes. Of course, the pair of sex chromosomes are the X and the Y chromosomes. XX, in the case of humans, means that you have a female. X and a Y chromosome mean that you have a male. The gametes are haploid, as I mentioned a moment ago. So the pair of chromosomes that we see here is going to split in meiosis, and the egg is just going to have one homologue, one part of this homologous pair of chromosomes. At fertilization, those will come back together. We'll have one from an egg and one from a sperm, and so the organism that's created will be diploid as a result of that union. So let's move on and talk about some other details here related to meiosis. Okay, so before meiosis, there is an interphase, nearly identical to the interphase that we saw in mitosis, and during this time DNA is duplicated. This means that when we enter meiosis, we see that all the chromosomes consist of two sister chromatids. Now that'll be familiar to you if you think back to our discussion of mitosis earlier in this chapter. Meiosis is different than mitosis because it takes place in two stages. As you see in our little diagram here, we have the interphase, G1, S, and G2, and at the end we have duplicated chromosomes, and then we have meiosis 1 and meiosis 2. We're going to talk about what happens in meiosis 1 and meiosis 2 over the next couple slides here. Meiosis 1 is responsible for separating members of a homologous pair of chromosomes, as we see depicted in our screen here in our little picture down at the bottom. So here we see what we're calling prometaphase 1, and in prometaphase 1 we see the homologous pairs of chromosomes are lined up at the metaphase plate. So the place I'm indicating with the cursor here is the metaphase plate, which is that imaginary line in the middle of the cell at which whatever's going to divide lines up. In mitosis, that's the chromosomes themselves. In meiosis, that's the homologous pairs of chromosomes, as we see here. So that'll be an exam question that you could, of course, anticipate. What lines up at metaphase, or prometaphase, of meiosis 1? And you will say homologous pairs of chromosomes line up, because it is in meiosis 1 that homologous pairs separate. We see that occurring right here. Now you may ask the question, well, why is this chromosome mostly red and a little bit blue? But we'll deal with that in just a moment, actually in the next lecture. That's a result of crossing over, and we talk about crossing over a couple pages from now in the chapter, so hold on for just a few minutes for that. Meiosis II is very similar to mitosis, because in meiosis II, what's going to occur is the chromosomes themselves are going to line up along the metaphase plate, and the sister chromatids are going to separate. So we can see all this occurring here in this diagram from our text. So here we have the end of G2, which is, of course, the end of interphase, as you know. Now, here we have prophase 1, and so in prophase 1, the chromosomes condense. They're found there in the middle of the cell. We then see metaphase of meiosis 1, metaphase 1. And so, again, that question that we can anticipate on the exam, what lines up along the metaphase plate, the line that I'm indicating right here with the pointer, the imaginary line in the middle of the cell, what lines up along the metaphase plate in metaphase 1, or metaphase of meiosis 1? And you will say homologous pairs of chromosomes, because it's going to, in anaphase, the homologous pairs are going to separate. So it is meiosis 1 that takes the cell from diploid to haploid. Remember, being diploid means that you have 2 copies of each chromosome. If you're haploid, you have 1 copy. And so here we're haploid. Now, there are 2 chromosomes there, but remember, these are not 2 pairs. So if you go back to this previous slide, maybe let's just go back for a second. What we see is that we have a big chromosome here, and so there's 2 of these, and they match in length there, and a smaller chromosome here, 2 of them that match in length. They're going to separate here, so this cell is diploid, 2 copies of each of these. We go to the next slide, and we see that these cells are now haploid. We have 1 copy of each chromosome, 1 big one and 1 small one. Now, this is a very simple cell, just for the purposes of teaching, that only has 2 pairs of chromosomes, 4 total. So remember, humans have 23 pairs of chromosomes, 46 total. So, of course, if this were a human cell, we'd have 23 chromosomes in this cell and 23 in this, not the 46, which is the diploid number. Now, this is the end of meiosis 1. We go to meiosis 2. So we have prophase 2, we have metaphase 2, and again, a good question for yourself, see if you can answer this, is what lines up at the metaphase plate in metaphase 2, or metaphase of meiosis 2? And the answer is, you should be able to say the chromosomes themselves line up, because in anaphase 2, the sister chromatids that make up each chromosome, and if you are a little uncertain about what sister chromatids are, then you can go back to the last lecture and review, or just remember that the sister chromatids are 2 halves of the duplicated chromosome. The sister chromatids are exactly the same, with the exception of what resulted from crossing over that we talk about in the next slide here, or beginning of the next lecture. But the sister chromatids are identical with that exception, and so they are pulled apart here in anaphase of meiosis 2. At the end of meiosis, we have 1, 2, 3, 4 separate cells. And these cells are haploid. So this is different than mitosis for several reasons. Mitosis gives us 2 cells. Meiosis gives us 4. Mitosis gives us cells that are diploid. Meiosis gives us cells that are haploid. It's also interesting to note that if you're creating sperm in this manner, you end up with 4 viable sperm that can go on to fertilize an egg and form offspring. But if you are creating eggs, then you only end up with 1 viable egg and 3 what are called polar bodies. Polar body is a way of getting rid of the extra DNA. And so, in other words, you can envision 1 egg here and then just this little tiny structure that contains these chromosomes, and the same thing here and there. Those are polar bodies. The reason for that is that eggs are big and take a lot of energy and materials, and so all of that has to be devoted in just to 1 egg. You can't afford to make 4 eggs, during this process. But when you make sperm are much smaller, don't require as many parts, and so 4 sperm result from meiosis, the process of meiosis. So again, this concludes our discussion here of meiosis. In the next lecture, we go on and talk about some things that happen during meiosis that contribute to genetic variation.